Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So in today's class, I will show you some of the advanced nanomaterials which are used for cellular imaging purposes. So if you remember, in one of the previous class, we already discussed about the available fluorescent proteins and the organic dyes which are being frequently used in cellular imaging applications. So in today's class, on top of those fluorescent proteins and the organic dyes, you will learn the availability of some nanomaterials or the nanoparticles which are being used for cellular imaging applications. So let us start with what are nanoparticles. Okay. So nanoparticles are extremely small particles of matter that are between 1 to 100 nanometer in diameter. Okay. So just to put you in reference with the size of the protein, generally the size of the protein is 1 to 10 nanometer. Okay, and these nanoparticles or the nanomaterials generally ranges in the size from 1 to 100 nanometer. Okay. And nanoparticles can be made from a variety of materials, including metals, ceramics, polymers, and biomolecules. And their small size give them unique physical, chemical, and biological properties which are different than their bulk material. So here I have given one example. So graphite. Okay, most of you must be knowing what the graphite is. Okay, we use the pencils which are made up from graphite. Okay, so graphite can be turned into nanodiamonds by breaking up graphite's atomic structure using high energy sound waves. Okay, so you just make the smaller particles from this graphite, and those are known as nanodiamonds. And these particles have the size in the range of 1 to 100 nanometer in diameter. Okay, so these nanodiamonds, so here you can see the size of the nanodiamonds, it's around 4 to 5 nanometer. Okay, so this is actually made from the graphite only. Okay, but because of its small size, it has the unique physical, chemical, and biological properties which are different than graphite. Okay. Now, which are the key features of these nanoparticles? So they have the high surface area. Because of the small size, they have high surface area. Okay, so that enhances their reactivity and interaction with other substances. Then the quantum effects. So at the nanoscale, the quantum mechanical effects can dominate, leading to unusual optical, magnetic, and electrical properties. And the tunability of the properties. The nanoparticles can be engineered to exhibit specific characteristics such as fluorescence, conductivity, and catalytic activities. Okay. So most of the nanoparticles that you can fine tune its properties. Okay, by several ways that we will learn in later part of this lecture. So here I have shown some of the typical nanoparticles. So they are known as carbon dots, quantum dots, polymer dots, modified silica, AIE dot. AIE stands for aggregated induced emission dot. Okay, aggregation induced emission dot. Then nanodiamonds and the upconversion nanoparticles. I will show you what is the meaning of this upconversion nanoparticles in the next slide. So these are different types of nanoparticles, and here is the size comparison of this nanoparticles. So you can see they ranges from 1 to 100 nanometers. These nanodiamonds are the smallest. They are just 4 to 5 nanometer in size. That means they are basically smaller than our proteins, because proteins are generally 1 to 10 nanometer in diameter. So nanodiamonds are maybe even smaller than the proteins, whereas carbon dots, quantum dots, they are more or less the same size of the protein molecules, and the polymer dot, modified silica, they are even higher than the protein molecules. Okay. So which kind of nanoparticles have been used in bioimaging? So several types of nanoparticles have been used for imaging applications, such as nanoparticles made from silica and organically modified silica, hydrophobic and hydrophilic organic polymers, semiconducting organic polymers, quantum dots, carbon nanoclusters and nanotubes, nanodiamonds, upconversion nanoparticles, metal particles, metal oxides, and many more. Okay, so there are several types of nanoparticles or the nanomaterials have been used for biological imaging applications. applications. Now here I will just explain you this terminology. What is the upconversion nanoparticle? So upconversion nanoparticles are the type of nanoparticles which generally absorbs the near infrared light and emits the UV visible light. See, most of the other nanoparticles like quantum dots or carbon dots, they absorb the UV visible light and they emit the near infrared light. Okay. Whereas these upconversion nanoparticles are generally kind of a two photon. They need two photon illumination, so they need a higher wavelength for illumination, near infrared, and they emit the light in the visible or UV range. Okay, so see, they can convert multiple low energy photons into a single high energy photon, the two photon illumination. And they can convert near infrared photons into visible light photons, which results in less autofluorescence and photo damage to biological tissues. And the composition of the lanthanide ions and the host material can be varied to produce a wide range of colors. Okay, so you can actually modulate the color output or the emission of the upconversion nanoparticles by changing the composition of the lanthanide ions and the host material it is made up of. Okay. Now, what are the benefits of using nanoparticles for fluorescence imaging compared to fluorescent proteins and fluorescent dyes? Okay, so you know there are so many varieties of fluorescent proteins and fluorescent dyes are available in the market, and they have been extensively used. Then why one should consider using the nanoparticles for cellular imaging applications, right? So you know that what is the major disadvantage of fluorescent proteins and even the fluorescent dyes is that they photobleach quite easily. Okay, and for your long-term imaging applications, they are not photostable. Okay, most of the time they photobleach, and also the brightness is little weak compared to those things. The brightness of this nanoparticles is much higher. Also, the nanoparticles provides high photostability. Okay, even some of the nanoparticles, they do not bleach forever. Okay, the nanoparticles are also inert to their microenvironment. So if they go inside the cell, then they will not interact with other cellular components, or they will not non-specifically bind to other cellular components. Even distribution inside the cell, most of the time they they are evenly distributed inside the cell instead of making aggregates and all. Some of them do make aggregates, but there are some ways of surface modifications by which you can avoid the aggregation of the nanoparticles inside the cell. And one of the most important point is here, the nanosensors have calibration plots which are quite similar between in vivo and in vitro systems, whereas the fluorescent proteins are quite different in that respect. Okay, so what it is telling, that if you make some calibrations or some measurements in vitro using the fluorescent nanoparticles, and when you inject those fluorescent nanoparticles inside the cell or inside the tissue, okay, the calibration plots remains the same. So basically their optical properties will not change whether the nanoparticles are outside the cell or they are inside the cell. Whereas in case of fluorescent proteins, their optical properties will change. If you are making some calibration curves in vitro system, the same curve may not valid for in vivo system, because in vivo environment, the optical properties of the fluorescent proteins will change. Okay, so that way the nanomaterials or the nanoparticles provide you quite comparable results between the in vivo and in vitro systems. Now, nanoparticles can be their properties can be modulated by several coatings and surface modifications, as they have a high surface area, right? So you can provide some coatings or the surface modifications to change their properties. So coatings and surface modifications of nanoparticles serve two purposes. The first thing is to make the nanoparticle fluorescent. Okay, because most of them are by nature they are non-fluorescent, but you coat them with some kind of a fluorescent dye or so that they become fluorescent. Okay, and to improve the properties of the nanoparticles. And which kind of properties? If you want to make them brighter, you want to make them more biocompatible, if you want to make them more cell permeable, because see, for visualizing anything inside the cell, you have to make them cell permeable. Okay, so you can make them more permeable by coating their surface with some kind of a polymers or so that they can easily go inside the cell. And target specificity. Suppose you want to make the nanoparticles which can specifically go and bind the cancer cells, or you want to make the nanoparticles which can specifically go inside the cell and bind to some specific proteins. You want to coat it with antibodies. Okay, so those kind of coatings can be done to nanoparticles to achieve the target specificity. Now, what can be attached to the nanoparticles by physical adsorption or covalent binding? So, what you can attach to the nanoparticle? Okay, so polymers such as polyethylene glycol to improve the uptake by the cell and protect nanoparticles from the immune system. Okay, so if the nanoparticles are coated with the polyethylene glycol, your immune system will not consider it as a foreign molecule. Okay, then you can also coat the nanoparticles by proteins or the antibodies, some fluorophores or like organic dyes, oligomers or other particles. And what are the desired properties for bioimaging? Like why you will think of coating or surface modifications to the nanoparticle to achieve which kind of properties? To make them more water soluble, to increase their biocompatibility so that the cytotoxicity can be reduced, because some of the metal nanoparticles, they are made up of heavy metals. So sometimes they may cause some cytotoxic effect. So to reduce that cytotoxic effect, you will coat their surface with some other biocompatible molecule so that the cell will not recognize that this nanoparticle is actually made up of some heavy metals, but it will only recognize the outer coating of the nanoparticle. Okay. Then you will do the surface modifications for increasing the cell permeability, increase its brightness, and specific targeting to cell organelles or cells or the biomolecules. Okay. So here are some examples. So gelatin was used to coat the CDTE or CDSE quantum dots to make them hydrophilic, more biocompatible and suitable for bioimaging. So see, this quantum dot is made up of heavy metal. So if you put directly this quantum dot inside a cell, it will create cytotoxic effect because of the heavy metal. Okay, so you have to coat this metal based quantum dots with a gelatin so that it will be more biocompatible and suitable for imaging. Then the cell permeability of the particles is strongly improved if the surface is coated with poly-L-lysine. Okay, to make your nanoparticles cell permeable, you should coat them with a poly-L-lysine. Then nanoparticles that can target cancer cells. Now for specific targeting, you see this example I will show you here. The rhodamine coated nanoparticles of 30 nanometer in size or in diameter are highly biocompatible by coating the surface with phosphocholine and lectin. Okay, so see, this is the nanoparticle. Okay, and they loaded rhodamine derivative so that it will give red color fluorescence, and then they coated this surface with phosphorylcholine and active ester groups. Okay, so that these nanoparticles have high affinity for the sialic acid overexpressed on the surface of tumor cells. So tumor cells generally overexpress the sialic acid on the cell membrane. Okay, so these nanoparticles have high affinity to bind to the sialic acid because they have the phosphocholine and lectin. So this way, these nanoparticles were made for specific targeting of the cancer cells. So you can see, if you treat the cells with these nanoparticles, then it will give the fluorescence when the cancer cells are present, but in the case of non-cancer cells, it will not give the fluorescence. Okay. Here another example, the coating of nanoparticle with triphenyl phosphine group causes the nanoparticles to accumulate near mitochondria. Okay, so this is kind of a mitochondria targeting group, triphenyl phosphine. Okay, so they just coated the nanoparticles with this TPP. It is known as TPP group. Okay, so these nanoparticles, you can see that they can be used to stain the mitochondria. So here what they have done, dual color imaging. So MitoTracker is the commercially available dye for identifying the positions of the mitochondria. Okay, so wherever we are seeing green color, those are the mitochondria in the cell. and this is a commercially available dye which is specific for mitochondrial labeling. So when they use these nanoparticles, so ideally these nanoparticles also accumulate near mitochondria. Okay, so the red color signal that you are seeing, those are the locations where the mitochondria are present. So you can see they are completely co-localizing, red and green. That means these nanoparticles are specifically labeling mitochondria because they are coated with this triphenyl phosphine group, TPP group. Okay. So this is how you can do the surface modifications of the nanoparticles, and you can specifically target any cell organelle or any specific cell type like the cancer cells versus non-cancerous cells, or even you can label any specific protein by coating them with a specific antibodies. Okay, so all these kind of surface modifications are possible with nanoparticles to either increase their biocompatibility, to either increase their cell permeability, or to make them specifically target any protein or cell or tissue in the within the cell or outside the cell. Okay. In this table, you can see the comparison of different nanoparticles for fluorescence imaging. So for here you can see these are the different nanoparticles: carbon dots, quantum dots, polymer dot, modified silica, aggregation induced emission dot, nanodiamonds, upconversion nanoparticles, carbon nanotubes, and metal based nanoparticles. Okay, and you can read this table for your reference, but I'll quickly go through some of the major advantages and disadvantages offered by different nanoparticles. So for carbon dots, for an example, they are water soluble and biocompatible. Okay, and they can be easily, their surface can be easily functionalized or modified, and they are quite photostable. Okay, but the disadvantage is that lack of red and infrared emission. So these carbon dots are available only in from green or violet range, but they are not available for the red emission. Okay, and broad excitation and emission bandwidth. So the excitation bandwidth is broad, so it can be excited by multiple wavelengths. Okay, and that's why if you want to do 2 or 3 color labeling with carbon dots, it's not possible because they have a broad excitation, right? Let's say they can be excited from 390 to 500 nanometer wavelength. Okay, so in those things, even our fluorescent dye, there are 2 or 3 dyes which can be easily excited. Okay, so they cannot be used for multicolor imaging, but one color you can image for a longer time because they are photostable. Then quantum dots. So they have a tunable particle size and surface modification, tunable PL emission. So PL stands for the photoluminescence. Okay, so they have the tunable photoluminescence emission and narrow emission band and high PL quantum yield, and they are photostable. Okay, so they are also photostable, and their emission bands are narrow, but how about the excitation? So here it must be mentioned broad absorption band. Okay, so their excitation band is broad, and risk of multiphoton excitation, and short off state times, and potential toxicity of heavy metal, because these quantum dots are made up of heavy metals. So the cells may show some kind of a toxicity against those heavy metals. And short off state time, what does it mean? So in some of the next slide, we will see that some of these nanoparticles have been used for super resolution imaging and STORM imaging, that is a stochastic optical reconstruction microscopy, that we already studied in one of the previous class under the super resolution imaging technologies. Okay, so for STORM imaging, if you remember, you need a dye which gives you the blinking property. Okay, the dye should blink, switch on and switch off for STORM imaging. Okay, so these quantum dots, good thing is that they offer this kind of blinking, but they're short off state times. Okay, so they blink just transiently, and that is not ideally suitable for STORM imaging. So you need to modify those things using some kind of a surface modifications. Okay, that we'll see in the next slide. Then the polymer dots, they have the large particle size. Okay, so sometimes they cannot be easily go inside the cell because of the large size, but they can be easily functionalized, bright and photostable, and they provide continuous fluorescence as well as photo blinking. So you can use them for storm imaging also, because they can give photo blinking also, or they can provide continuous fluorescence. And potential toxicity of degradation products. So some of the degradation products may be toxic for the cell. Then modified silica, so it is biocompatible, easily surface modifiable, then enhanced fluorescence effects, and they are also photostable. And the disadvantages are relies on doped fluorophores. So this modified silica doesn't have its own fluorescence. Okay, so the fluorescence is always dependent on the doped fluorophores. Means you have to add some, you have to modify the surface with some fluorophores. And potential toxicity of the degradation products. Similarly, you can just follow all these other types of nanoparticles, their advantages and disadvantages. Okay. Now the next question is how to target the nanoparticles to specific cell organelle or cells or biomarkers. Okay, because generally for biological imaging, we need some kind of a specificity so that the nanoparticles can specifically label either some proteins or some cell organelles inside the cell. Okay. So internalization is facilitated by modifying the surface with polyethylene glycol and or coating with the cell penetrating peptide, which also avoids endosomal sequestration. Okay. The nanomaterials functionalized with DNA aptamers have been applied to targeted imaging of specific sites inside the cell. So you can just refer to this paper in case if you are interested in going into details of this. And the tumor cells are best recognized by targeting their folate receptors. It was targeted with an aggregation enhanced fluorescent silica nanoprobe and used for both 1 photon and 2 photon microscopy. Here is the reference in case if you want to go into the details. And here I have provided this table, and this table is from this review article, Volpaste at 2015, and he has nicely summarized all the applications of fluorescent nanoparticles for targeted imaging. And this reference numbers that mention those actually refer to this publication. Okay, so if you are, let's say, interested that how the gold nanoparticles modified with hairpin DNA to target mRNA was used, you just go to the 45 number reference of this review article. So you can go into the details that how they made this kind of nanoparticle for specific targeting of mRNA. You can just see here quantum dots functionalized with streptavidin and the zwitterions used to track the cannabinoid receptor in live cells. Okay. Then graphene oxide nanosheet modified with an aptamer and carboxyfluorescein used for intracellular molecular targeting. So these are different ways people have targeted nanoparticles for specific biological applications, and whatever finds interesting to you, you can just go into the details of those by following these references. Okay, so now we will discuss about the applications of nanoparticles for super resolution microscopy. Okay, so as in one of the previous class, I already explained you 3 major types of super resolution imaging technologies known as SIM, STAD, and STORM. Okay, SIM stands for the structured illumination microscopy. Then STAD stands for stimulated emission depletion microscopy, and the STORM stands for stochastic optical reconstruction microscopy. Okay, and we have discussed all these 3 techniques in detail in one of the previous class. So in case if you are not familiar, just refer to that previous class to get an idea about how this 3 super resolution imaging technology works. Okay, and all these nanomaterials have been extensively used for all these 3 types of super resolution imaging microscopy: carbon dots, quantum dots, polymer dots, modified silica nanoparticles, aggregation induced emission dots, nanodiamonds, and upconversion nanoparticles. Okay. Now, in upconversion nanoparticles, I have mentioned here that they are not good for storm imaging. Okay, why is this so? So, if you may remember that for the storm microscopy, you need a fluorescent dye that blinks. Okay, so photo blinking or the blinking of the dye is very essential for storm microscopy. If a dye do not blink, if it gives continuous fluorescence, that is not good for storm imaging. Those kind of dyes are good for SIM and STAD imaging, but if they cannot blink, then storm cannot be done using those kind of dye. So this upconversion nanoparticles provides the continuous fluorescence. They do not blink. Okay, so they are not good for storm imaging. Whereas the other types of nanoparticles, either they themselves they blink, or using some kind of a chemical modifications, you can make them blink. Okay, so we will see that which kind of chemical modifications are required to make this nano particles blink for which is suitable for storm imaging. Okay, so all these nanomaterials differ in their fluorescence and photoblinking properties. So different chemical modifications are necessary to make them useful for super resolution imaging. Also, several optical and imaging regimes were adapted to use them for super resolution imaging. So in the next slides, we will discuss about some of these nanoparticles. Okay, so let us start with the quantum dots. Okay, so quantum dots offer outstanding photostability and high fluorescence brightness. Then they are more than 100 times more photostable than rhodamin 6G dye, which is generally used for super resolution imaging. Okay, with an almost 20-fold increase in brightness. So these quantum dots, they are 100 times more photostable and 20-fold more bright than the regular organic fluorescent dyes such as rhodamin 6G. Okay. Quantum dots offer a very broad excitation spectrum, while fluorescence emission is sharply confined to a narrow band of wavelength. So here you can see these are the excitation and emission band wavelength for the quantum dots. So there are 5 different types of quantum dots, and you can see that their excitation spectra is quite broad and more or less the same. Okay, whereas the emission spectra is very confined, like for it confined for less than 50 nanometer. Okay, so in a way that is a drawback. So with quantum dots, you cannot easily go for multicolor imaging, because whatever the quantum dot you take, okay, it is going to get excited at the same wavelength. Okay, so there is no specific wavelength for exciting one particular quantum dot. Even if you, let's say you are using the light at 400 nanometer or even 500 nanometer, okay, all 5 of them will get excited and they will give the emission in different channels. Okay, so for multicolor imaging applications, it may be little problematic, but as long as you want to image just one or at max 2 colors, you can still somehow manage with the quantum dots. Okay, and here is the structure of the quantum dots is shown. This is the core, then this is the shell, and this is the ligands. So ligands, you can make them specific, you can coat them with some antibodies so that inside the cell they can go and bind to some specific proteins. So here you can see how the quantum dots have been used for super resolution imaging, and we'll see that for all 3 technologies, that how they have been used for STAAD. In the next slide, I will show how they have been used for STORM and SEA. So the high photostability of quantum dots make them promising candidates for STAAD imaging. However, the bottleneck is their relatively small stock shift. Okay, so what do they mean by the relatively small stock shift? Let us see the second sentence and then I will explain. The broad excitation spectra extend into their emission spectra. This increases the probability of fluorescence re-excitation by the STAAD beam. So you can see that the excitation spectra is merging with their emission spectra. Okay, and because of that, what happened generally for STAAD microscopy, if you remember, there are 2 lasers used. One laser is to illuminate your dye, means it will be anywhere from 400 nanometer or even 390 nanometer to 650 nanometer. That is an excitation laser, and the STAAD laser. So STAAD laser is generally in this range from 700 to 800 nanometer. Okay, near IR range. Okay, so the STAAD laser, which is known as the depletion laser, sometimes it may also illuminate some of the quantum dots. Suppose if you are using QD655, then STAAD laser can illuminate this QD655 because it can also get excited at this wavelength. Okay. There is also a possibility of free excitation by 2 photon absorption of light from the powerful STAAD laser. Now the STAAD laser, the depletion laser, is very powerful. Okay, and because of that, what happened? 2 photon absorption. So let's say the the wavelength of this stared laser is, let's say, more than 700 nanometer. Okay, but it can create 2 photon excitation of the fluorescent dye. And this factor has so far limited the potential of quantum dots in stared imaging. Okay, so what happens here in this picture you can see. So here there are 2 lasers on, excitation laser plus stared laser. Okay, so these are the real structures that ideally you should see by this imaging. Okay, but you are also see a halo around those dots. Okay, and those halo is because of the stared laser and the fluorescence re-excitation by the 2 photon absorption light. So the stared laser will excite the molecules around these, and when you only use the stared laser, you can see that the real structures are missing and only the halo you can see. So this is because of the 2 photon excitation by the stared laser. So in this study you can see Han et al. In 2015 used ZNS coated CDSE or CDTE quantum dots, that is known as QD705, to observe QD stained vimentin fibers, and a resolution of 106 nanometer was achieved. And to reduce the 2 photon re-excitation, what they did, they collected the images by keeping both the lasers on. So here you can see both the lasers are on, 628 nanometer and 775 nanometer. So you can see the real structure as well as the halo due to 2 photon excitation. And by keeping only the stared laser on. So here you can see only the stared laser is on. So you can only capture the halo and not the real signal or real structure. Okay, and then you can subtract this image from this one to get, so A minus B is this image. So this is the real stared image. Okay, and this is the confocal image to show you that how the resolution has increased. Here there are 3 particles, they look like this in a confocal microscopy, whereas using stared imaging you can see them nicely separated. Okay, then Yang et al. In 2016 they used the same quantum dot 705, but they used a continuous wave depletion laser at 775 nanometer. Okay, instead of a pulsed picosecond laser source that is more conventionally used for stared imaging. So generally for stared imaging, and in this study, the previous study, they used the conventionally used laser that is known as the pulsed picosecond laser, which is a very intense source of light. Okay, so they used. So by using this continuous wave depletion laser instead of this pulse picosecond laser, they could avoid this kind of fluorescence re-excitation, and the authors demonstrated a lateral resolution of 85 nanometer for the visualization of the microtubule network in HeLa cells. Then in another study in 2020, they demonstrated that green emissive CDSE at ZNS quantum dots, that is QD526, under 488 nanometer excitation could not be re-excited by a 592 nanometer continuous wave depletion laser. Okay, so here instead of using the quantum dots in the red range, they use the quantum dots which are green emissive. Okay, and under 488 nanometer excitation, and they used 592 nanometer depletion laser, and that is a continuous wave depletion laser, not the pulse picosecond traditional laser. Okay, that is generally used for storm imaging. So they use this continuous wavelength depletion laser at 592 nanometer, and that they mentioned that using this illumination strategy, they could not see the fluorescence re-excitation. So they could not see this kind of halo with this kind of laser, and when they use this quantum dots in the green range. Okay, and on a single quantum dot, they achieved a resolution of 21 nanometer for this single quantum. Okay, so this is the highest resolution of 21 nanometer they could obtain using this quantum dots. Now, how the quantum dots have been used for storm imaging? So as such, the quantum dots show blinking, okay, which is useful for storm imaging. However, the blinking rates are fast compared to the traditional dyes that have been used for storm imaging. So this blinking behavior of quantum dot is strongly affected by the quantum dot structure, shell thickness, the presence of trap states, surface ligands, and the external environment. Okay, so the blinking rate can be modulated by changing the structure of the quantum dot. So in this study, the wide field and storm images of quantum dot 565 stained microtubules and quantum dot 705 stained mitochondria in HEP G2 cells. This is the image for the same. You can see the wide field imaging and storm imaging. Here they've used the quantum dot 565 to stain microtubules in blue color and quantum dot 705 to stain mitochondria in red color. Okay, so this is a storm image. You can see it is much better resolved compared to the wide field image. And the resolution of 24 and 37 nanometer were achieved in lateral and axial dimension. So in XY, they achieved the resolution of 24 nanometer, and in Z, they achieved 37 nanometer resolution. And due to fast blinking property of the quantum dots, they are good for super resolution optical fluctuation microscopy known as SOFI. Okay, so this is slightly a different type of microscopy, but it is mainly based on storm imaging only. Okay, and it only collects the optical fluctuations. Okay, so it is more or less like the storm imaging only, but it requires the fast blinking of the fluorescent dye. So the microtubules were labeled using the secondary antibodies conjugated to quantum dot 625 and visualized by SOFI imaging in 3T3 mouse embryonic fibroblast cells. Okay, so you can see that in any cell line, you can conjugate the quantum dots with secondary antibodies, and then you can visualize what are the protein of your interest. Okay. And for SEAM imaging, the quantum dots have also been used. 3D SEAM was used to image quantum dot labeled CD13 protein on the surface of single acute myeloid leukemia cells, revealing the distribution of individual protein on the cell membrane. And the QD605 was used to record both the distribution and the density of integrin receptors on single acute myeloid leukemia cells in this study. So if you are interested, you can just go to these publications and see the details at how exactly the microscope setup was used for SEAM imaging. Then let us come to the carbon dots for super resolution imaging. Okay, so carbon dots show excellent water solubility, low cytotoxicity, good biocompatibility, and unique optical properties. They can be functionalized via linker chemistry for targeting biomolecules. And CDs were conjugated with lauryl betaine BS12, a compound with antibacterial property. The BS12 modified CDs, it is denoted as CD C12, can be used for detection and inhibition of gram-positive bacteria such as Staphylococcus aureus. Okay. The CDC12 enabled bacterial imaging with STED and provided threefold resolution enhancement compared to confocal microscopy. A cationic carbon quantum dot probe emits spectrally distinguishable fluorescent signal upon binding to DNA and RNA in live cells. So this is very interesting. So this is known as a cationic carbon quantum dot, and it gives different color fluorescence when it binds to DNA or RNA. So when this quantum dots binds to DNA, it gives green color fluorescence, and when it binds to single-stranded RNA, it gives red color fluorescence, thereby enabling real-time imaging of DNA and RNA localization and motion. The probe can penetrate through various types of biological barriers in cells and in vivo for super resolution microscopy and time-lapse imaging of chromatin and nucleoli during cell division and in C. elegans growth. Okay, so here you can see this is a C. elegans germ cells. Okay, and this dye, it binds to DNA and RNA and it gives different fluorescence. So during cell division, where the DNA is there, chromosomes are there, you can see they are stained in green color, whereas the RNA molecules, they are stained in red color. So this kind of differential staining is also possible with the use of carbon dots. Now they have been used for storm imaging as well. So microtubules inside HeLa cells immunostained with primary and secondary antibodies conjugated with carbon dots. So here you can see the storm image, and this is the conventional fluorescence. So here you can see the structures more clearly. the enlarged portion of both this figure is shown below here. Okay, so this is a kind of a standard immunofluorescence protocol that was followed to stain microtubules inside HeLa cells, and they obtained the resolution of 60 plus or minus 6 nanometer, and fixation was done using 1:1 acetone methanol for 10 minutes. This was the blocking 6% BAC and 10% goat serum was used, and primary antibody cells were incubated with mouse anti-alpha antitubulin antibody, and the secondary antibody, the CD coated with goat anti-mouse IgG secondary antibody. Okay, so this is a quite common practice that you can coat any secondary antibody on your nanoparticles, quantum dots or carbon dots. Okay, so how to coat these carbon dots with secondary antibodies? So here is this brief procedure. So carbon dots in methanol were allowed to react with EDC and NHS and mixed with goat anti-mouse IgG secondary antibody, so that the secondary antibody will be coated on the surface of the carbon dots. Now, limitations of carbon dots for super resolution imaging. So most CDs have emission wavelength in blue to green spectral region, and the availability of CDs emitting in red and infrared spectral regions would be highly desirable. The broad excitation bandwidth of CDs makes multiplexed applications difficult. So as I showed you that the excitation spectra is quite broad for CDs also. So multiplexing is quite difficult. Most of the applications of CDs are reported for unspecific labeling. Okay, CDs that can be targeted to specific intracellular locations, target biomolecules are needed. Okay, so in most of the applications, CDs have been used for non-specific or the unspecific localization. Okay, but targeting CDs to some specific biomolecules, some proteins or some cell organelles is quite difficult. Although here we saw one application in which you can coat your carbon dots with secondary antibodies and use them for imaging specific proteins inside the cells. Okay, but that is in fixed cells. But in live cells, if you want to target those carbon dots to some specific cell organelle or to specific protein, so those kind of applications are limited. Okay, so now coming to the polymer dots for super resolution imaging. So the diameter of the polymer dots ranges from 20 to 30 nanometers, and compared to the molecular dyes, polymer dots offers high brightness and high photostability, just like the other nanoparticles. They can provide continuous fluorescence as well as photoblinking behavior depending on their composition. And polymer dots over 15 nanometer in diameter gives continuous fluorescence, whereas they are below 10 nanometers, the same material exhibits photoblinking. Okay, so it is also size dependent as well as the composition dependent. The smaller the size, less than 10 nanometer, the same material gives photoblinking, and if the diameter is greater than 15 nanometer, then it gives continuous fluorescence. So dual color study was performed to understand the dynamic interaction of clathrin derived endosomes and caveolin-1 positive endosomes in HeLa cells with a resolution down to 68 nanometer. Okay. Then STEAD imaging was performed in live cells to track the movement of endocytic vesicles labeled by biotin conjugated polymer dots. So you just have to observe that how the polymer dots can be conjugated for specific targeting. Okay, so here they conjugated polymer dots with biotin for specific targeting to endocytic vesicles, and here they use this clathrin derived endosomes and caveolin-1 positive endosomes in HeLa cells. Then the single molecule localization microscopy, means the STORM imaging, was performed using spiropyran conjugated PFBT, and conjugation with streptavidin permitted the specific labeling of microtubules and membranes in live MCF7 cells. And streptavidin conjugated PFBT and CNPPV have been used to label and visualize mitochondrial membranes, nuclear pores, microtubules in BSC1 cells. Okay, so here you can see these are two polymer dots, one for green fluorescence, one for red fluorescence, and here are the confocal and STEAD images of both the polymer dots. You can see that in the STED images you can resolve the structure better. And here, using the same two polymer dots, the the lysosomes were labeled using CN-PPV, and the CD44 was labeled using PDF-DP polymer dots using antibodies against CD44. And you can see both the lysosomes and this CD44 protein under the confocal as well as STED imaging. And you can see the improvement in the resolution. This is the comparison of photostability. So this FONPs are the polymer dots. Okay, and you can see that they offer a very good photostability. Over 25 minutes of time, they do not bleach at all. Whereas if you compare it with the regular fluorescent dyes like Elexafluor 594 or FITC, they bleach much faster than these polymer dots. Here is the excitation spectra. So they show a broad excitation spectra. Let's say if you take this polymer dot, that PFPT plus PFDBT5, then it can be excited at almost any wavelength in the visible spectrum. Okay, and so generally this is a common problem with most of the nanomaterials, that they are excited, that they have a broad excitation spectrum. But again, the emission spectrum, you see, it is relatively narrower than the excitation spectrum. Okay. Then coming to the nanodiamonds for super resolution imaging. So the nanodiamonds are inherently biocompatible and they are exceptionally photostable. Their fluorescence lifetime is longer. It's 20 nanosecond, which is much longer than the biological autofluorescence. The nanodiamonds can be surface functionalized to realize the specific labeling and targeting functions in the cells, and hence nanodiamonds are fascinating alternatives to organic dyes and fluorescent proteins and quantum dots. They are available in both red and green emitting wavelength, and they can be used for two-color imaging. And they have been used for two-color imaging by STED microscopy. And nanodiamonds have been used as dual contrast probes in correlative STED and transmission electron microscope imaging. So see, this is known as the correlative microscopy. So the same structure can be visualized by electron microscopy for getting the highest possible resolution, and the same sample is imaged under the STED imaging so that you can see the fluorescence. Okay, so this is the same image of the same sample which was imaged using the STED microscopy, and here you can see that in detail. So there is the electron micrograph, and it is superimposed with the STED image of this area, and you can see the structure in detail along with the fluorescent molecules with high resolution. So this is known as correlative microscopy, and these nanodiamonds can be used for this way correlative microscopy as well, because they can be seen by the electron microscope also. So apart from its several applications for super resolution imaging, the nanoparticles are used for imaging pH, imaging oxygen, imaging temperature in the cells or in the tissue samples. Okay, so I will show you some examples how they can be used for sensing pH. So here is one specific example, that two kinds of polystyrene particles, one being pH responsive because of the fluorescent pH probe immobilized on its surface, and the other containing an inert reference fluorophore, were applied on a wound. Okay, so this is a wound sample, and in which two types of probes were used for the surface modifications of the polystyrene nanoparticles. Okay, so the one probe is pH responsive, and because the fluorescent pH probe was immobilized on the surface, and the other probe is just used as a reference. Okay, so you can see the image here, and here there is a pseudo color image of pH values on an inflamed wound, and the bluish color indicates fairly intact skin where the pH is between 5 and 6. Okay, whereas the green and red color represents the areas of sustained inflammation and partial granulation. So here the pH ranges from 7 to 9. Okay, So this is how the nanoparticles can be used for seeing the pH differences in the wound sample or any tissue sample. Okay, and there are several other studies in which the fluorescent nanoparticles have been used for sensing intracellular pH values. So you can see all these references are from this review article. So the reference number 61 represents the 61 number reference in this review article. Okay, you see there are several other good examples like gold nanoparticle pH sensor based on ratiometric photo-induced electron transfer and confocal microscopy to image localized intracellular pH. Okay, then pH sensitive CDSE, ZNSE, ZNS quantum dots used to image pH value in human ovarian cancer cells. So in different types of nanoparticles have been designed to quantify pH at the subcellular level. Similarly, several different kinds of nanomaterials or the nanoparticles have been developed for imaging the oxygen concentration. So here I will show one example, and the other examples are summarized in this table from this review article. But let us see this example. So nanosensors were developed from polystyrene containing two luminophores matching the red and green channel of the digital color camera. Okay, and the red emission of the oxygen probe is quenched by oxygen. So in this tissue sample, wherever the oxygen is present, okay, the red fluorescence is quenched. So you will not see the red fluorescence. Okay, but where you are seeing the red fluorescence means that the oxygen level is less. Okay, and they use this another dye, that is blue-green emitting reference fluorophore. So this is a green color dye that you can see. It's a reference fluorophore. It is not sensitive to oxygen concentration. Okay, whether the oxygen concentration is high or low, it will remain green only. But this red dye is a oxygen sensor. So if the oxygen concentration is high, it will quench the red emission. Okay, and here is a ratiometric image. So here you can see the black means the highest oxygen concentration. Black areas shows the highest concentration, whereas the orange, yellow, or even white, those areas represent the lack of oxygen. Okay, so these areas in this tissue sample, these are the areas where the oxygen is not there, whereas the black area represents the high amount of oxygen. Okay. So this is how the nanoparticles can be used for quantifying the oxygen level in the tissue sample. Okay. Similarly, the nanoparticles can be used for imaging temperature. So here you can see a fluorescent polymeric thermometer was developed, and that this thermometer is nothing but actually it's a nanoparticle, and it can diffuse throughout the cell, and it can be used for intracellular temperature mapping, where the fluorescent lifetime of this fluorescent polymeric thermometer is measured as a temperature dependent variable. Okay, so see, this thermometer contains these three elements: thermosensitive unit, hydrophilic unit, and the fluorescent unit. Okay, so so under the cold condition, it gives weak fluorescence, but upon heating, the three-dimensional structure of this changes, and it gives this fluorescent dye some fluorescence. Okay, so this is a strongly fluorescent molecule when at high temperature, but again upon cooling, it can go back to this open conformation, which is weakly fluorescent. Okay, so using this thermometer, you can see fluorescent intensity at 564 nanometer, and it can detect the temperature from 20 degree to almost 50 degree. Okay, it gives this kind of a curve. So as the temperature increases, the fluorescence increases. Okay, and here you can see the temperature mapping in living COS7 cells by fluorescence lifetime imaging. So this is the cell line in which these nanoparticles were injected, so that you can see that nanoparticles will go inside the cell, and based on the temperature, they will give fluorescence. Okay, or fluorescence lifetime actually. Okay, so you see what they observed, that there is a temperature difference between the cytoplasm and the nucleus. So in the cytoplasm, the temperature is 33.1 degree, whereas in nucleus, the temperature is 35 degree. Okay, so it's a 2 degree temperature difference in different cell organelles, like in cytoplasm versus nucleus. Okay, And there are some other types of nanoparticles have been developed for temperature sensing, which are listed in this table. Also, the nanoparticles can be used for bimodal or multimodal imaging. Okay, so what is a bimodal or multimodal imaging? So here you can see one example. In this mouse system, you wanted to see that some tumor in fluorescence as well as in CT scan. Okay, so these nanoparticles are designed in such a way they provide both the properties, the fluorescence as well as the properties required for the CT scan. Okay, so this is a typical design of a multi-layered dual modality OFI or CT for OFI or MRI nanoprobe. Okay, so the functionalized layer 1 and 2 are for attachment of fluorescence reporting agent or conjugation with additional biocompatible and targeting group. Okay, so see, this is the core of the nanoparticles, that is iron oxide based MRI contrast agent. Okay, so this core will act as a agent for CT scan. Okay, whereas this functionalized layer 1 fluorescent reporting agent, functionalized layer 2 and biocompatibility layer and the targeting groups, these are the targeting groups will take these nanoparticles to specific site, okay, wherever the tumor is or wherever some specific cells or tissues are there. Okay, whereas these other layers are required to make them biocompatible, to make them functionalized, to make them fluorescence. Okay, and this core is required for the MRI or CT scan. So bimodal or multimodal imaging is equals to combination of fluorescent imaging with other methods of imaging such as magnetic resonance imaging, plasmonic imaging, and positron emission tomography, PET imaging. Okay. Depending on the kind of multimodal imaging, a second species is required to be present in the nanoparticle in addition to the fluorescent probe that will give a signal in MRI or PET. So it is saying that for multimodal imaging, see, this is the layer which gives fluorescence. Okay, the dark pink or magenta layer. It's a fluorescent reporting agent. But you need another chemistry for to be detected by the CT or MRI. Okay. In this study, OFI, that means optical fluorescence imaging, and CT, computed tomography probe, was designed as shown below, and this probe could detect the tumor site in HepG2 tumor bearing nude mice in vivo with either near infrared fluorescence or CT imaging. Okay. So this is how you can make the nanoparticles or to modify the nanoparticles for dual modal imaging. You can combine the fluorescence imaging with other type of imaging technologies like MRI, CT or PET imaging. Yeah, so that's it for today. And for this particular lecture, you should follow these two review articles. Okay, if you need to go into the details of any one of these applications, just refer to these review articles, and then you will get some more references from there which you can follow. And we'll see you in the next class. And in the next class, we'll discuss about the immunofluorescence and immunohistochemistry. Thank you.